Hello, my lovely anatomists and physiologists, and welcome back. I have taken us back in time a little bit to cardiac output, just to kind of act as our placeholder, a reminder of where we've been and where we're going. So cardiac output, remember, is talking about, how effective of a pump is your heart? How much blood is your heart pumping out each minute? And remember, this is a factor of stroke volume, which we spent a couple of videos talking about, and heart rate. And so heart rate is actually where we're ready to pick up our conversation now. So heart rate, remember, can be measured by your pulse, where you can actually feel the movement of the blood as it's pumped through the radial artery. And it is the number of beats per minute. So we're seeing like how many cardiac cycles can your heart do in a minute. And what we'll see is that our average heart rate for an adult is about 75 beats per minute, but that this can range. This can, of course, range. So we do see like a healthy range of 60 to 100 beats per minute. Now, heart rate is something that is being controlled by nervous system, endocrine system, and local factors. And so what we're looking at here is the nervous system control of the heart rate. And so I have this diagram that I've pulled off of Google, where you can see the brain, and we have neurons located, remember, in the medulla oblongata, which is the bottom part of the brainstem becoming continuous with the spinal cord. And what we see is that we have two different regions. We have what's described as the cardio-accelerator region and the cardio-inhibitory region. Or instead of the word region, we can use the word center. So you could do either term here. And what we see then is we have either sympathetic division neurons or parasympathetic division neurons that are then synapsing onto those conducting cells of the heart, or what we can call myocardial conducting cells. Those are the cells of the pacemaker system. So remember, we have those cells of the heart that have their own spontaneous depolarization rates. But here, what we're talking about is an override to that spontaneous depolarization. And what we'll see is that the medulla oblongata has what are called chemoreceptors that are going to be responding to if we have high carbon dioxide gas levels, if we have high hydrogen ion levels. Actually, let me change this and say we have the chemoreceptors that are responding to concentration of carbon dioxide gas, concentration of hydrogen ion, and concentration of oxygen gas. So we're looking at all three of these. When we talk about the heart rate, we will see that if you increase your heart rate, you're going to increase your cardiac output to a point. Because remember, we've also said increasing filling time will increase stroke volume. And so these two things are at odds. Meaning if your heart is beating too fast, you don't have adequate filling time, which means you're reducing your EDV, which means you're reducing your stroke volume, right? But it may be that your heart is beating fast enough to sort of compensate for this reduction of stroke volume. So we see this little bit of a compensation that happens because these are multiplied against each other. If you reduce stroke volume and increase heart rate, you maintain good cardiac output. If you increase stroke volume and reduce heart rate, you can still maintain good cardiac output. So there's sort of like this optimal range for your heart rate. So when we say we can increase our heart rate, increase cardiac output, that's as long as we stay within this range of like 120 to 160 beats per minute. If we really go faster than this range, then we're not getting enough filling time and we're beating too fast to really, even compensate for this reduction in stroke volume, right? And so we'll see a problem with that. So we will see there's kind of like this balance that has to exist here. When we talk about cardio acceleratory center or region, of course, this is increasing heart rate. It's in the name, cardio accelerator. And so of course this means, it's the sympathetic division. And what we see is the sympathetic division here is going to release norepinephrine, which is a neurotransmitter / hormone that we've talked about throughout the semester. And what we'll see is that the norepinephrine increases your heart rate because it reduces the, it reduces the repolarization of the cardiac muscle. And then it increases the depolarization of the cardiac muscle. And so collectively, you're increasing your heart rate. So what that means is you are allowing the cardiac muscle to be stimulated sooner than it was otherwise. And so it can contract more quickly. And so you're increasing your heart rate. So what it's going to do, the norepinephrine is going to do this by actually opening up what we can call chemically gated sodium ion and chemically gated calcium ion channels. And so we're slowing down, we're reducing the repolarization period, and we're bringing on the depolarization faster. We are seeing that the norepinephrine is binding to beta-1 receptors. And so sometimes patients are prescribed beta blockers in order to slow down heart rate if you're having trouble regulating your cardiovascular system. When we look at the cardio inhibitory center, this is of course the opposite. So now we're slowing down the heart rate. So this would be the parasympathetic division. The parasympathetic division, we know, is releasing the neurotransmitter acetylcholine. And acetylcholine is going to do the opposite. So we're going to slow down or reduce the depolarization. And then we're going to extend repolarization. So if we slow down the depolarization rate and extend repolarization rate, then the cardiac muscle cells are contracting more slowly. And we do this by opening up potassium ion channels. I should say chemically gated here. Chemically gated potassium ion channels. And so the potassium is leaving the cell, and so that's reducing the depolarization and extending repolarization. What we're going to see is that the parasympathetic division is going to dominate resting heart rate. So if you actually look back to our discussion of pacemaker system, we had the SA node sort of in charge, and we said that it depolarized at a rate of, I think, 80 to 100 beats per minute. But here we're saying that the average adult heart rate is 75 beats per minute. So that's actually slower than our kind of like in charge set of conducting cells. And so the reason it's slower is because of that parasympathetic division. What we'll see, we mentioned that the medulla oblongata has chemoreceptors that are sensitive to carbon dioxide gas concentrations, hydrogen ion concentrations, and oxygen gas concentrations. And so what we'll see is when we talk about our cardio accelerator region, this is going to be stimulated when you have high level of CO2, high level of hydrogen ion, and low levels of oxygen. And this is typical of an active tissue. So particularly if your skeletal muscle is active, then you're seeing that the cells are using up a lot of oxygen, producing a lot of carbon dioxide gas as a waste product. And we also will see the buildup of hydrogen ion. So release of hydrogen ion is an indicator of active tissue. And so if this is the case, then we need to increase our blood that's moving around the body, right? If we're having a lot of active tissue. And so we'll see this from activating our cardio acceleratory region. So then we can expect to see the opposite in the case of our cardio inhibitory. So when our tissues are at rest and not very metabolically active, we get a reduction of hydrogen gas, we get a reduction of carbon dioxide gas, a reduction of hydrogen ion, and you have more oxygen present in the body. And so this is more a mark of resting tissues. There's one other important center that's present in our medulla oblongata. So I'm going to grab a blue color here. So we're still going to be talking about here in the medulla oblongata. So remember, we had the cardio accelerator center or region. We had the cardio inhibitory center or region. And then we have what's called the vasomotor center. And the vasomotor center is controlling what we call like a vessel tone. So we're seeing that our blood vessels are going to have a smooth muscle, and the smooth muscle is going to stay a little bit constricted all the time. And this little bit of constriction is coordinated by the vasomotor center. And what we'll see is that this control of the vessels is dominated by the sympathetic division. And what we'll see then is we get a little bit of tone all the time, so that if we need to constrict the vessels, we're going to increase our stimulation, more contraction, constrict the vessels. And if we go back to kind of our regular tone, if we need to dilate the vessels, you're going to suppress, you're going to stop sending signal, and you're going to allow for relaxation. And so that's going to allow that vessel to dilate. So also when we talk about having elevated carbon dioxide gas and elevated hydrogen ion, we'll also see here, in addition to stimulating the cardio accelerator system, this will also stimulate the vasomotor center in order to get vasodilation. Or the opposite of that, when we have our reduction in CO2 gas and our hydrogen ion and our increase in oxygen, then we're going to suppress the vasomotor system in order to allow for vasoconstriction. Okay. That's it for this conversation on heart rate and looking at the controls that are located there in the medulla oblongata. Stay tuned for our next video. As always, take care of yourselves and each other.